Welcome you dear students. I do trust you are watching this video with your good health and happy mind. I feel privileged being part of Zoological Society of Assam. This endeavor in disseminating knowledge to students as per state's undergraduate zoology curriculum at the time of COVID-19 outbreak. Well, we will discuss today about nucleosides and nucleotides. I wish to highlight the importance, or you can say the significance of nucleoside and nucleotide by saying these are parts of DNA. I intentionally first use the term DNA because the word DNA has crossed the boundary not only of biology but also science. Every other person who is not into science yet knows that there is something called DNA, which is a carrier of genetic information. Every other person without biology or science background may not know what exactly RNA or nucleoside or nucleotide is comparatively, but DNA has gained such popularity. That is the reason a simple double helical structure is enough to pass a scientific flavor to the common audience, be it is a movie of Spider-Man or Jurassic Park. And this is the one of the reasons I fell in love with my subject zoology. It says the truth of life. This negates any kind of discrimination on the basis of caste or creed and at the same time validates the diversity due to geographical locations, climatic conditions, food habits, etc. etc. I read zoology a bit, so I am saying so. But every branch of science destination is reaching the ultimate truth, be it as a physical science or the chemistry inside ourselves. Even the arts and philosophy depicts that eventual truth in a beautiful way. That's the reason Dr. Bhupen Hazarika sang Manu He Manuhar Babe, a man for humanity, and Anil Pawan Barua finds meaning of life through his paintings. Anyway, I say it negates the discrimination. Therefore, be it genes, me, or a plant, when it comes into a blend of two, it's just a permutation and combination of four letters ATGC. These letters are the nucleotides. How? Let us know in the upcoming few slides. So after this presentation, we are supposed to know what is nucleosides and nucleotides, how to call the nitrogenous bases in it, what is the purines and pyrimidines, tautomerism in them, sugars in nucleotides, and their names, formation of nucleosides and nucleotides, and of course with the take-home messages. The nucleotides came into existence with the discovery of nucleic acid by Johannes Frederick Misser from the nuclei of past cells from discarded surgical bandages in the year 1869. He reported he had found something in the nucleus of WBC, which is slightly acidic in nature, and named it as a nuclein. But he couldn't determine its function at the time. Few years later, he is successfully able to separate the components of nucleic acids, that is, nucleotides. It was Richard Altman who coined the term nucleic acid to nuclein in the year 1889. He termed it so because of its discovery in nucleus and the presence of phosphoric acid in it. Well, there is an apology for breaking a small rule. We generally go for the functions at the end. Let's have the function bit early here to understand why we need to know about nucleotides. As already mentioned, they are the monomers which repeated many times form polymers of DNA and RNA. But this is not the only functions they do. They have a role in energy currency, as you have heard about adenosine triphosphate or ATP or GTP. They also act as a precursor of several important coenzymes such as ionic NAD, ionic NADP, FAD coenzymes. They act as a precursor of secondary messengers like cyclic AMP. They also serve as an intermediate in some biosynthetic reactions. For example, S-adenosyl methionine in methyl transfer reaction. So I guess by the time we are convinced that nucleotides are important in biological system and we need to study this. With that belief, here we go. Nucleotides are made up of three components: a base, sugar, and an ion of phosphoric acid, phosphates. Let's go back one step. If we minus the phosphate from the nucleotide, it will become nucleoside. Now let us go one step forward. If we keep adding one nucleotide after another, it would become nucleic acid. This is the interrelation among nucleoside, nucleotide, and nucleic acid. We will restrict our presentation today up to nucleoside and nucleotides with their structural components. Let us know the bases first. The bases and nucleotides are the nitrogenous base, that is bases that contain nitrogen atom. The nitrogenous bases in nucleotides are aromatic heterocyclic compounds. They are ringed structure. There are two types of nitrogenous bases, purines and pyrimidines. Again, purines is of two types, adenine and guanine. On the other hand, pyrimidines are of three types, cytosine, thymine, and uracil. Now, how to call them, the purines and the pyrimidines? Purines are numbered anti-clockwise, while pyrimidines are numbered clockwise, as per an internationally accepted system to represent the structure of bases. Thus, in case of purine, which is double ring or bicyclic structure, starting from here, numbers will be 1, 2, 3, 4, 5, 6, 7, 8, 9. Please do not get confused with the numbering 7, 8 and 9. One can assume this ring on the other side and then call 1, 2, 3, 4, 5, 6, then 7, 8, 9. This structure is made for convenience of representation only. So we have to name the two purines, adenine and guanine. Following the rule of numbering, we can see an amino group at the sixth position of the ring of adenine. So as per the numbering 1, 1, 2, 3, 4, 5, 6. So a amino group in the sixth position. The name will be 6-aminopurine. So please remember, we generally call him adenine like your nickname, but specifically this is 6-aminopurine. In case of guanine, you can see an amino group at the second position. Again, please compare with the numbering system. So in the second position there is amino group, and at the sixth position there is an oxy group. Therefore, the guanine is actually 2 amino and 6 oxypurine. Now let us move to pyrimidines. As I said, this is to be named in clockwise direction. So starting from here in the clockwise directions, the number would be 1, 2, 3, 4, 5 and 6. No extra ring present here unlike purines, they are monocyclic. As you know by this time, there are three types of pyrimidines: cytosine, thymine and uracil. Let's name them. In case of cytosine, as per the numbering system, we can see one oxy at the second position. Please compare with the numbering system. So one oxy group in the second position and one amino group at the fourth position. So the other name of the cytosine would be 2-oxy, that means 2-oxy and 4-amino pyrimidine, because these are pyrimidines. 2-oxy, 4-amino pyrimidines. In thymine, there are 2-oxy group at the second and fourth position. Second and fourth position, 2-oxy group, and a methyl group in the fifth position. Thus, it would be called 2,4-dioxy because at the position 2 and 4 there is a 2-oxy group, thus 2,4-dioxy, and 5 in the position 5 there is a methyl group. So 2,4-dioxy 5-methyl pyrimidine. Let us see uracil. Here, same like thymine, 2-oxy groups are present at position 2nd and 4th. But of course there is a no methyl group unlike thymine. Thus the name would be 2,4-dioxypyrimidine. Molecules with keto and enol forms exhibit tautomerism. The heterocyclic rings of purines and pyrimidines with oxo-functional group shows tautomerism. Let us see the example of uracil. These undergo shifts from keto to enol form. Ring nitrogen serve as a donors of hydrogen bond, and the keto oxygen behave as hydrogen bond acceptor, giving rise to keto-enol tautomerism. The keto form is called lactam and the enol form is called lactine. So keto is a lactam and enol is a lactine. Like uracil, guanine, cytosine, thymine do also exhibit the tautomeric character. I believe you can guess why adenine doesn't have the tautomeric characters. Because of lacking of oxo-functional group in it. But guanine, cytosine, thymine and uracil having their oxo group in them. The keto-enol tautomerism, the equilibrium avoids any nuclear instability causing loss of formal aromaticity of the molecule. At a normal physiological condition, lactam or keto form is predominant. So by now, we have got the nitrogenous bases in our hand: adenine, guanine, thymine, cytosine and uracil. However, there are some minor base or unusual bases are found in nucleotides such as 5-methylcytosine, pseudouracil, N4-acetylcytosine, N6-methyladenine, etc. This is a general prediction that the unusual bases in nucleic acids help in recognition of specific enzymes. Let us shift from base to sugar in nucleosides and nucleotides. The sugar in nucleoside or nucleotides are 5-carbon monosaccharides, thus called as pentose sugar. More specifically, there are two types of sugar, beta-D-ribose sugar and beta-D2-deoxyribose sugar. Beta-D-ribose sugar is present in RNA, while beta-D-deoxyribose sugar is present in DNA. In regular term, we called ribose sugar in RNA and deoxyribose sugar in DNA. Let us see how they are different between them. As I already said, sugars in nucleoside and nucleotides are pentose sugar. So 5-carbon sugar, carbon 1, 2, 3 and 4 are within the ring and carbon 5 is outside the ring. The carbons in sugar is represented by prime attaching with it. Thus carbon of pentose sugar are 1 prime, 2 prime, 3 prime, 4 prime and 5 prime that is outside of the ring. May I please have your attention at the carbon number 2. In deoxyribose of DNA, at this carbon position 2, there is a lacking of 1 oxygen. Thus this is called 2-deoxy. 2 means at the position of carbon 2 and deoxy means lacking of 1 oxygen. So ribose sugar in RNA and deoxyribose sugar in case of DNA. Let us move into a very interesting topic, that is a sugar ring puckering. The sugars prefer difference pucker in nucleic acids. This is called sugar puckering. The four carbons at the four corners form a plan of envelope. The oxygen is at the position representing the top of the envelope. The oxygen can be bent out from the plan of that envelope. Twisting the carbon at 2' at 3' position in relative to other atoms give rise to two different Parker forms of sugar. Twisting of carbon at a 2' position from the plane of the 4 carbon give rise to C2', that is carbon 2' endo conformation. Similarly, twisting of carbon in the 3' position from the plane of the 4 other carbons gives rise to C3' or carbon 3' endo conformation. So now we have got sugars in addition of bases in our hand. So if you can recall the initial explanation, so if we have a base and sugar, then this is definitely a nucleoside. So we have made the nucleoside till now. But what would be their nomenclature? Based upon the nature of the sugar, the nucleosides would be called ribonucleosides and deoxyribonucleoside. If the base is A, then the ribonucleoside would be adenosine. Similarly, if the base is guanine, it would be guanosine. If the base is cytosine, it would be cytidine. If the base is uracil, the ribonucleoside would be uridine. Similarly, the deoxyribonucleosides would be deoxyadenosine for adenine, deoxyguanosine for guanine, deoxycytidine for cytosine, deoxythymidine for thymine. In case you have noticed, uridine is only present in ribonucleoside. And similarly, The thymidine is present only in case of deoxyribonucleoside. This is because uracil is present only in case of RNA and thymidine is present only in case of DNA. Thus, uracil is linked with the ribose only and thymine is linked with the deoxyribose only. So, if I say mere thymidine in case of deoxythymidine, it would represent the same thing because there is no point mentioning deoxy in prefixing thymidine as by default thymine always part of deoxyribose only. This can be present in ribonucleoside. So we came to know base plus sugar give rise to nucleoside. But how this attachment occurs? The base and the sugar get attached by glycosidic bond. More specifically, this is a beta N glycosidic bond. The bond is said to be beta because of its up configuration with respect to the ribose sugar. Let us see the bonds in purine and pyrimidine. This is the sugar and this is the base. Here, this is the sugar and this is the base. Let us recall the generic structure of sugar and the bases. So here you can see this is the glycosidic bond through which the sugar and the base is attached. So let us match with the generic structure of the sugar. We can see this is the carbon position 1 of the sugar and this is the position 9 of the base. You can assume the ring on the other side and then you can easily understand this is the 9th position of the base. So in case of purine, the bond is between C1 prime of pentose sugar and N9 position of the purine ring. So more simply between 1 prime and 9. The 1 prime is to denote this is from the carbon 1 prime position of pentose sugar and 9 is the position of the base. Similarly, in case of pyrimidine, this is corresponding to the 1 prime position of the sugar and this position is again the 1 position of the pyrimidine base. Therefore, in pyrimidine, the bond is in between carbon 1 prime of pentose sugar and N1 position of the pyrimidine ring. So more simply, it is between 1 prime and 1. This 1 prime is to denote this is the carbon 1 prime position of pentose sugar. In the sugar-base conformation, the base has a property of free rotation over the glycosidic bond. As a result, two different conformations are possible known as syn and anti. You can visualize the conformational changes in the purine rings giving rise to syn and anti conformation due to the rotation of the base over the glycosidic bond. In case of pyrimidine, which is single-ringed or monocyclic, adopts only anti-conformation. So, syn and anti for purine and only anti for pyrimidine. Now let us add a phosphate to it and we'll get the ultimate nucleotide. Nucleotides are the phosphoric acid esters of nucleosides. The first phosphate primarily attaches with the sugar at the 5 position. Please recall the generic structure of sugar. So this is the 5th position, 5th carbon out of the ring of the sugar where the phosphate is attached. The nucleotide can have 1, 2, 3 phosphate group known as alpha, beta, gamma for the first, second and third attachment as you can see attachment of 3 phosphates that is phosphates. Addition of phosphate in nucleoside sometimes can form two ester bonds at the 5' and 3' hydroxyl group of the ribose sugar. This forms a cyclic nucleotide. For example, let us visualize in cyclic adenosine monophosphate. So this is the position of 5' and this is the position of 3'. They play important role, the cyclic nucleotides play important role in regulation of metabolism. Their role includes protein-ligand interactions and act as a secondary messengers in hormone and ion channel signaling in eukaryotic cells. Therefore, now we are having base plus sugar plus phosphate. Thus, now we have nucleotide in our hand. Next would be how to call them. Again, based on the sugar, there are two types of nucleotides, ribonucleotides and deoxyribonucleotides. For base adenine, the ribonucleotide would be adenosine monophosphate because we are saying about unit nucleotide. So, one phosphate or monophosphate, one phosphate attaches to the 5' of the sugar. So, all are 5' monophosphate. This is also called as adenylate and abbreviated as a AMP. For guanine, the ribonucleotide would be guanosine monophosphate or guanylate, abbreviated as a GMP. For cytosine, the ribonucleotide would be cytidine monophosphate or cytidylate or CMP. For uracil base, the ribonucleotide would be uridine 5' monophosphate or uridylate, abbreviated as a UMP. In case of deoxyribonucleotides for base adenine, it would be deoxyadenosine monophosphate or deoxyadenylate, abbreviated as a small d for deoxy-AMP. For guanine, it would be deoxyguanosine monophosphate, that is 5' monophosphate, or deoxyguanylate, and abbreviated as small d GMP. For cytosine, it would be deoxycytidine monophosphate or deoxycytidylate, abbreviated as small d CMP. For thymine, the deoxyribonucleotide would be deoxythymidine monophosphate or deoxythymidylate, abbreviated as small d DMP. This is all same to abbreviate DTMP or simply TMP because thymidine always part of deoxy, that is deoxyribonucleotide. Well, you have seen in DNA and RNA sequences only the base sequences are shown. For example, if you are familiar with NCBI or any other such public databases, you have seen such sequences. For those who are not much aware of NCBI, an example and a link is shown below. You can visit the database NCBI. You need to select the nucleotide in the drop-down box of the database, and if you put any accession number, for example, if you put accession number of this, you would be able to see the nucleotide sequences. Here we have shown one of our DNA sequences that we have obtained from a fish called Anguilla bengalensis and submitted in the NCBI databases. Going there, you can see the stretches of ATGC in different combinations. You won't get any uracil because that is a DNA. Can you guess why only the nucleotides are shown every time, not the sugar or the phosphates? Because the change in the sequence occurs only due to the change in the position of the nitrogenous bases, ATGC. sugar and phosphate backbone remains constant. At the starting, I did mention about the truth of life. The truth is the permutation and the combination of these nucleotides, more specifically the basis of the nucleotides. The ATGC in all deoxy one after another link together and form nucleic acid DNA. Similarly, AUGC in different combinations join one after another to form the RNA. The condensation between two or more nucleotides occurs most commonly in the alcohol of the 5' phosphate of one nucleotide and the 3' hydroxyl of the second, with the elimination of a water molecule forming a phosphodiester bond. So as per the topic of today's presentation, I do restrict here up to nucleotide. Subsequent presentations later will explain the structure of DNA and RNA in details. Before I do sign off, the take-home messages. Nucleotides are the building blocks of DNA and RNA. Besides, they have a role in the formation of energy currency, precursor of enzyme, precursor of secondary messenger, activated intermediates in biosynthetic reactions. Nucleotide minus phosphate is the nucleoside. There are four primary nitrogenous bases in nucleic acid: A, G, C, T or U. Purines and pyrimidines with oxofunctional group show ketoenol tautomerism. The base rotating over glycosidic bond while attaching with sugar exhibit syn and anti conformation. Depending upon the nature of the sugar, there are two types of nucleosides or nucleotides: ribonucleoside and ribonucleotides, and deoxyribonucleosides and deoxyribonucleotides. Twisting at the 2' and 3' carbons form different markers in the sugar conformation. Thank you indeed for listening. You can pin down queries over here. Stay safe. Thank you very much.